first thing that we're going to talk about is protein synthesis. Protein synthesis is to synthesize means to make, and protein obviously means protein, so to make proteins. And the first step in making proteins is a process called transcription. So the pathway to making proteins starts in the nucleus, and to do that we go from DNA to mRNA. And then we modify our mRNA to just the portion that we need. This is the process of transcription, and it takes place in the nucleus. From our modified mRNA, it's going to leave the nucleus and go to the ribosome where it's going to make proteins. This is the process of translation, and this occurs in the cytoplasm, more specifically at the ribosome. This is basically an overview of what we will do in the next two videos. So we're going to talk about this process of going from DNA to RNA to protein. This happens in eukaryotic cells, and if you take a look at this diagram, you will see that we have a nucleus. We go from DNA to this pre-mRNA, basically mRNA that has some extra information in it. And then we go from there to mRNA that's going to actually leave my nucleus. Everything that's happening inside my nucleus is transcription. So transcription specifically goes from DNA to mRNA. After it leaves the nucleus, it goes into the cytoplasm, specifically to the ribosome. We're going to take that mRNA strand and translate it into our new language, which is proteins. So what does it mean to transcribe something? Well, if we think about it, this is a doctor speaking into a recorder. Sometimes you will encounter this when you go to the doctor's office. The doctor then gives the recorder to a medical transcriptionist who will type up the doctor's notes after listening to the recording. This is transcription. They are both in the same language. The doctor speaks into the recorder, and the transcriptionist listens to it and basically puts it onto paper. Again, both in the same language. So, the process of copying a sequence of one strand of DNA, the template strand, into mRNA is the process of transcription. Both are in the same language, mainly nucleotides. So although some of the bases in my DNA and RNA may be different, they are both made up of nucleotides. This requires the enzyme RNA polymerase, and we know what the function of an enzyme is. What is the function of an enzyme? If you were thinking to speed up a chemical reaction, you would be correct. So RNA polymerase is going to speed up the reaction of making RNA. So question, what would be the complementary RNA strand for the following DNA sequence? C, GC, GT, ATG. Well, think. When I'm making RNA, what pairs with G? If you were thinking C, you would be correct. For my next base, what pairs with C? If you were thinking G, you would be correct. It gets a little tricky when we get to our T's and A's. What pairs with T? If you were thinking A, you are correct. So let's take a look at what the transcribed sequence of DNA would look like. It would look like C, G, C, A, U. Notice instead of a T pairing with an A U is pairing with an A. Because remember, we are in the process of making RNA. So the process of transcription has some steps. The first step is that the enzyme RNA polymerase binds to my DNA and separates the DNA strands. Next, free-floating nucleotides form bonds with the DNA strand building the mRNA. More enzymes remove unnecessary parts of the RNA molecule before they move out of the nucleus. So the part that's going to actually code for my DNA is going to be the exons. The introns are going to be removed. After after the DNA is transcribed into RNA, editing must be done to the RNA to make it work. Introns are my non-functional pieces, basically my junk, and they are snipped out of the RNA. Basically, we want this process to be as efficient as possible, so we don't need to send any junk out of the nucleus into our cytoplasm. So if we take a look at this, you'll notice that your DNA is made up of orange pieces and blue pieces. The blue pieces are the pieces that actually code for genes, and the orange pieces are the junk pieces. We will copy that entire strand, the junk and the good pieces, to make an mRNA. Then we will take out all the junk pieces and put the good pieces together, and that gives me the actual mRNA that's going to go out of my nucleus. So notice all of the orange pieces are taken out because they're junk. They are called introns, non-coding regions of the DNA and RNA. Exons are the pieces of DNA that code for proteins. They are rejoined by enzymes. So let's try this. Write the complementary nucleotide for the RNA strand. So here's my DNA, A, G, C, T, T, C, G, A, G. My RNA would be U, C, G, A, G, C, U, C. Notice, anywhere where I would think a T would be, I've put a U in. So here's my DNA strand, and I'm going to write my mRNA strand for it. First, I need to take my DNA strand and transcribe the entire thing. So everywhere I see a T in my original strand, I'm going to put an A. Everywhere I see an A, I'm going to put a T. Everywhere I see a C, I'm going to put a G. And everywhere I see a G, I'm going to put a C. The next step is to take that strand and put it into mRNA. Notice the pink line. These are my mRNA. The shaded nucleotides represent introns that will be cut from the mRNA molecule. The remaining parts of the molecule are the exons that will be spliced or cut and glued together by enzymes to create my final mRNA. So everywhere I see a shaded region, that part is removed from the sequence. So down here, I'm going to, in my next step, copy down just the letters that are doing coding. Notice A, U, G, A, C does coding. The next part got cut out, the A, U, U, it did not code for anything. So then I put the G, G, C, U in. I cut out the next part and I put in U, A, G because that's the next coding region.